All right, welcome everybody to our Data Science Zoominar. Today we are very fortunate to have microbiologist and scientific integrity consultant, Dr. Elisabeth Bik. Dr. Bik is known for her work detecting photo manipulation in scientific publications. That's what we're going to be talking about today. And I want to have one announcement is that today we have the Office of Research Integrity joining our Zoominars for the first time. It might be the first time they come to one of our academic talks. So welcome everybody from that office. I think you might learn something today. All right, so Dr. Bik, I'm gonna start asking you some questions. The first one is that first I wanna explain that this is an audience of data scientists. Many in the audience is quite familiar with errors in the literature introduced by incorrect data analysis. And this has been particularly problematic in genomics where a lot of us work. And part of it is because there's a lot of untrained individuals analyzing big data-driven studies led by PIs that some of them aren't really experts in data analysis. So in fact, in our field, there's this new area called forensic bioinformatics. We have a couple of famous people, Bagley and Coombs, that were instrumental in exposing a problem in an important paper from a group in Duke that eventually became known as, I think, if you Google Duke cancer trial scandals, you see it on the internet. And it was basically a long process to make that come out, in part because it was based on data. Many of the editors didn't necessarily understand what statisticians were saying. But you find problems in images rather than data analysis. Yeah. And that's different. That's a little bit different. And I think biologists are a little bit more understanding of that and are more easily convinced about that. So can you give our audience a brief intro on what you do and how you became interested in this area?
Yeah, sure, of course. Thank you for having me here today. So I did my PhD in microbiology, but I've worked 15 years at Stanford. And there I got interested in looking at images just by listening to podcasts and hearing and reading some data on science misconduct. And I got immediately grabbed by that idea, and I first focused on plagiarism. But later I discovered that I have this weird talent to spot duplicates in images. So I decided to scan the literature as a hobby project in my free time, to scan biomedical papers for duplicated images within that paper. And I thought I wouldn't really find a lot, but I found immediately some examples. And so I just scanned first 10 times.
Can I interrupt you? So yeah, when you say you saw you had a talent, how does that happen? Like you show it to your friends and you're like, did you see this? And they're like, what? I don't see anything. And then you're like, look, is that how it kind of happened? Like, how do you notice that you have a special talent for it?
I was looking at a PhD thesis that had plagiarized text, and I just flipped through the images, and there was this photo of a Western blot, a protein blot, basically a photo with a little smudge, a little stain, and a couple of pages later, that same photo appeared, and it was upside down. So it was rotated, and it represented a different experiment. And I'm like, hey, I've seen that image before, and that paper had been published. So I figured nobody had seen this. This was peer reviewed. It was published. It was cited by others. And there's a big error in it, or maybe it is not an error. I don't know. But I just decided to scan papers, and I found so many that first night that I thought I must have this talent because they're all peer reviewed, right? Like all these papers have been seen by others, and so why didn't the peer reviewers or the editors or the readers see this? So I must spot this.
I see. Interesting. But let me interrupt you real quick, and just to let the audience know, this is being recorded. It's going to be on our YouTube channel later in a week, maybe earlier. So I'm getting questions online about that. So yes, you'll be able to listen to it later. Okay, continue. Sorry.
Okay, so I also have this, I guess, the same talent to spot duplications in bathroom tiles. So if I go to a bathroom in a hotel or in a restaurant, and sometimes they have these patterns patterned tiles on the wall or on the floor, and I'm just bothered because I see the patterns. I'm like, that tile is the same as that tile, or but that tile is rotated or so. So I've always seen that, and I assumed other people did the same. But I guess I'm pretty unique in that. But by posting these things on Twitter, so now I'm discovering that other people are starting to see that as well. So it's one of those things. If you point them out, then people start to notice it too.
Yeah, definitely. And yeah, so for those that don't know, you can follow Dr. Bik on Twitter, and she puts up challenges, and she tells you the level, like hard, easy, medium. you can try to answer them. Okay. So are you gonna show us some slides now, or you want to do that later?
Sure, I can show some slides. So
yeah, so people know what we're talking about.
Yeah, all right. Let's share my screen. And that's this one. So I'll present. So you can see my screen, it's building up. There it is. So this is a paper I published together with Arturo Carcedoval and Ferric Fang in 2016. And this is sort of my big work in which I looked at images in biomedical papers in a sort of a systematic way. I looked at over 20,000 papers. So I didn't read the papers, I just looked at the images. And I I selected these papers by screening for a particular word called Western blot, which is a type of photo, a type of image in these types of papers, to enrich for papers that have photos. Because I didn't look at line graphs, I focused on the photos in these papers, and I want to enrich these papers for photos.
And since this is one of the questions I had is, how did you pick these 20,000? Is it all of them, or anything with a Western?
No, I started by scanning PLOS ONE, so I can show that.
Okay.
Let me see, I'll move around. So this is the study set. So I started by scanning papers from PLOS ONE, which is a sort of middle of the pack journal in terms of impact factor, but it's a wide journal. They'll publish a lot of different topics. And I specifically scanned for the word Western blot between quotes, and scanned a couple of months worth of papers from 2 years, 2013 and 2014, which is when I did this work. And then later I decided I should not just focus on one journal, I should look in other journals as well. So I added another bunch of journals and also went back in time. So this is why the study set is a bit lopsided, and in terms of numbers of papers from PLOS ONE. But it's a very easy journal to flip through. It's not because I have some particular suspicion about PLOS ONE, and in the end it actually ended up in the middle of the pack in terms of percent of image duplications. But it's just very easy. You just load. It's open access, so I can just do this at home, flip through the images in a very fast time to have like detailed view of images. It's just very nicely set up for the type of work that I wanted to do, which is to look at photos. So that's the data set. And just to go back in my slide, so I base my searches on the fact that in biology, research images are mostly unique. So I have a couple of examples here on the screen. On the top right are Western blots, which is the type of photo that I most look at. So they're black stripes, and for me all these stripes look unique. They have unique different shapes. They have different colors of background. There's little smudges and stains and just the shape. They have smiling. This one smiles a little bit, or it frowns a little bit. You have all these different shapes. So I just look at this image, and this is a great image. It's unique. So I'm specifically looking for duplicates in these things. So these are microscopy images of all kinds, and some of these images look similar to each other. This image looks similar to that, but it's actually a time series. And so you would expect these things to look similar. So even if a duplication was there, but it was appropriate because it was a time series, or it's the same experiment shown in figure 1 and in figure 5, if that's a duplicate, but it's showing the same thing, I would not flag it. I would only flag it if they're inappropriately duplicated, which means that it's a duplication, but the two identical images represent different experiments, which is not good, not expected. And so those are the ones I flagged. And here are some examples. This is what I call type 1 simple image duplications. For example, if you compare these two photos that are marked in red, and the colored markings, the colored boxes are mine. So I added them to the photos. They're not part of the original. So these two look identical, and these two, one here boxed in blue, also look identical. You see this little curved structure here that you see in both of these. Well, if you look at the labels, they're different experiments, so that's not good. And then here are some of those examples, and here are two as well. So those are duplicate panels or duplicated images.
And how long, when you look at this, how quickly do you see it? Like I only
within seconds.
I see it after you, within seconds, because I see it once you mark it, but otherwise, okay.
Well, I know what to look for. So so first of all, I will immediately think, oh, all these images look a little bit similar. So I'll look for a structure like this. Do I see this one somewhere else? And then I see it. And so I have, of course, after scanning 20,000 papers, I have some experience. So I'll know that, oh, this might take me maybe 1 minute or so, because I'm like, oh, is this the same as that, but rotated because it has a little white dot? No, it's actually a different image. And so, you know, some of these look quite similar to each other, like these three, but they are different. And so that might take me a little bit longer to spot it. And once I find one pair, there might be a second one, so I will scan a little bit more.
Okay.
And this is another type of duplication, but with repositioning. So it's either, you know, rotated, it's flipped, or flipped upside down, it's shifted. So all these are examples where, for example, these two panels here, they have a common area marked here in green by me. And so even though they're supposed to represent 4 panels, you actually see only 2 different photos. It's just a shifted.
So I'm sorry, but someone went into Photoshop, cut stuff up, and moved it around.
No, it's not photoshopped. This is just shifted. So the color boxes are mine. So it's just to show that these 2 images overlap. So this left corner of this panel, this top left corner overlaps. So they're sort of like that. Yeah. You just shift the microscope a little bit and take another photo.
Got it. It's like a different screenshot.
Exactly. Yeah. And here in this example, you'll see the same image, but it's flipped. You can see here the labels are flipped. So I had to rotate the whole image to make these 2. This is a Western blot. So to make these 2 lines or these 2 rows look exactly the same. So there's a little spot here that I labeled with a blue arrow. And similarly here, this is the same blot, but in the left one, it's labeled that it came from a nucleus of a cell, and it's the tubulin protein, while here it came from the cytoplasm, and it's the HDAC1 protein. So that's definitely not the case. These are the same blots, it's just shifted. So they're shifted. And then the third one, that's the baddest example. So this is what I think is image alteration or photoshopping. For example, here you see lane 9 and 10 look very similar to each other. And so it looks like it's just one photo, but it looks like part of the photo has been duplicated. And maybe lane 6 and lane 8 also have things in common that I see, like this structure here, you can see here. And here in this example, although this is not technically a photo, I know that, but these are called flow cytometry images, and I include them in my searches because I can spot the alterations there as well, and they're supposed to be unique. They're like observations plotted in a grid, and you see a lot of similarities between these 2 images. But here in the middle, you see a region that is not present here, and so I don't know if this has been added to the image or taken out in this image, but the percentage of dots that fall in these quadrants have been changed, and so this looks like a doctored image. Wow. So to recap, there's 3 types, and this third type is most likely to have been done intentionally, while the simple duplications are the most likely to be an honest error. So that's why we have these 3 categories.
So this last one, I don't know how to use Photoshop. I don't own it.
Me neither. I just spotted.
So how do you, do you know how to use it though? Like, is this easy to do in Photoshop? Because like taking points out, is that something you can like click on them and erase them? Like how easy is it
Yeah.
to produce this from? I can see what you're saying. These are the same with some either added or subtracted points. Yeah. But that, I guess maybe it is easier. Maybe someone in the audience can that knows Photoshop can tell us how hard that is to do. But anyways,
yeah, I mean, just look online. There's so many altered images nowadays. It might not be in the program Photoshop itself, but you could probably even add it in other programs like Paint or so. Whatever, like you just add a couple of dots, or maybe they had another image which looks similar but not shown in this particular figure, and they took a part of that cluster of dots and plopped it into this particular plot. So
someone in the audience says it's pretty easy. Yes, with brush and clone, it's easy. Everybody's
exactly,
like, it's super easy. Okay, thanks everybody. You got it.
I'm not very good in Photoshop. I've used Illustrator a lot, which is also Adobe program, but Photoshop no, I'm not very good at it. And but yeah, I think it's one of those things. If you know how to use it, it's pretty easy. And I assume it's easier than actually doing the experiment, because otherwise you would just do the experiment, right? But they didn't. They Photoshopped it.
Great.
So yeah.
Fascinating. Okay, so can I continue with my questions?
Of course.
Okay, so you have a statement in one of your papers that says the prevalence of papers with problematic images has risen markedly during the past decade. So what is that statement? That was in the intro, not in the results. So what is that statement based on? Is that on your own data, or is that something else you've seen that makes...
Yeah, we didn't put a reference there. It's just based on the observation that you see more and more in the news articles about papers that were retracted because of image problems. And it's also fed by the fact that a paper nowadays has more figures than 20 years ago. When I did my PhD, you would have one image or two images, and that was a really nice paper. But nowadays, images and figures in publications have much more panels, and there's more supplemental. So it's just the number of figures per paper has risen a lot. But yeah, there's websites like Papier or Retraction Watch. If you follow those websites, you'll see a lot of papers being retracted because of image problems. That's a substantial part, and it seems that is on the rise. But yeah, it's just fairly easy to spot these things in photos. And so you can even wonder, why do people leave that many traces? Because if I can see that overlap, why don't you shift the microscope a little bit farther, and there would not be an overlap, and I would not spot it.
Maybe they are. And
well, that's true. That's true. Yeah.
That relates to my next question somewhat, which is that your false positive rate seems to be very low. Like I don't think I've ever seen some image that you share that I think you're wrong. Mm-hmm. But how about do you have an idea? This is hard, but do you have an idea what your false negative rate is? Have you looked at an image, not seen anything, and then have somebody else tell you, oh, you missed this one? Like, do you have any sense of that?
I don't know a percentage, but I'm definitely not perfect, because the way I scan these papers is by scanning really fast. So it might just take me a minute per paper, depending on the complexity of the paper, obviously. But yeah, so I definitely miss duplicates that other people see. Sometimes it turns, when I post these things on Papier, there will be other people and saying, oh, you missed one. I found, like, let's say 20 duplicates in a photo, and somebody would say, yeah, well, I found the 21st one. And I'm like, okay, fine. But I miss things because I'm trying to do these scans pretty fast. I have no idea about the percentages. I might miss maybe, let's say, 30% if I had to make a wild guess. Wow. But on the other hand, you could say, sure, I miss these things, but all the people who have reviewed, edited, and cited the papers have not seen them. So I'm still finding many more than.
Absolutely, no, there's no question about that. And that brings me to a question from the audience, which is similar to one I was going to ask. I'll ask them at the same time. So have you had conversation with editors and if you have had them, are they listening? Do they realize that that they could be doing more? And the question from the audience is that, given that the current system isn't working as well as it would if there was like 100 people like you looking at these things, is there a possibility of having companies, for profit? The question is for profit companies that actually do this for every paper.
Right. That's a lot of questions in one question.
Yeah.
So the talking to editors, I haven't really talked one-on-one with editors a lot. I've had email conversations, obviously, because all these 800 papers that we found in this set, I've reported. But I think to really see if editors are listening is to look at the results, if these papers have been corrected or retracted. So after I reported these duplicates, which are inappropriate, so they're either errors or they're intentional. But either way, in my opinion, there should be either a correction or a retraction, depending on the severity of the duplications. So of the 800 papers I've reported in 2014 and 15, so that's 5 years ago or longer, only 30% of these papers have been corrected or retracted.
Interesting.
And so that means that 2/3 of these papers have not been touched. They're still out there, even though I reported them 5 years ago, which means that the editors are not taking any actions. They don't seem to be very motivated to look at these papers. And it's of course a lot of work for them. They need to contact the authors, ask for original data, and maybe the authors don't respond because their email address doesn't work anymore. And so they're, maybe stuck in the situation where they don't want to take any action, or they don't have the capacity, they don't have staff to deal with these things. So, but I think over time, now that I'm posting these examples on Twitter, that I'm sort of dragging it in the open and forcing people to take a look at this and making decisions if we are okay with these papers having duplicated images, or should we do something. So I feel the tide is changing a little bit, and that editors are starting to realize that this is a real problem, and that they should address it, and not only look at these things post publication, which is what I'm doing, but before publication. Right, sure. So that's the second question is, can we look for these things by some automated way? And I actually turned this into a drinking game. So I'm going to take a sip of coffee, because whenever I'm asked this question, I usually reply on Twitter by saying drink, because it's a drinking game. I get asked this question a lot. And so yes, there are companies and and also academic groups that are developing software tools to scan for these things. And because you would think it's fairly easy to scan for duplicated parts of images, but it's much harder than one might think. So there are now some tools on the market, or starting to come on the market, that I assume will be used by publishers to screen manuscripts that are submitted like they screen manuscripts for plagiarized text. So with plagiarism, they scan the text to all the papers that have been ever published and see if there's a duplicate. For images, that is a similar process, but the scale computationally is much larger. And so it's not quite possible yet, but I assume that's a matter of time. These tools are, I believe, still not as good as the human eye, because they look for pixel to pixel identity, while sometimes these things are not pixel to pixel identical, and they're rotated or shifted, or they might have a different color, or they're like stretched. And so with your eye you can recognize it, but sometimes the software doesn't catch these things. Yeah, well, we can't tell cupcakes from chihuahuas. Exactly. Like artificial intelligence is not always a good thing. And so I think you'll always need a human intervention or a human assessment to see if what a tool finds to be identical, if that's appropriate. Maybe it is the same experiment, or it's the same very similar blood because they reprobed the blood with something else. And so you'll always have to have a human looking at the results of these tools and say, yes, that's okay, or that's not okay. Interesting. So on a follow-up question to that is that you clearly have a talent for this, and
the question then is, can you think we can train people that aren't necessarily naturally gifted to do this? And I'm thinking of referees, like that maybe there could be a course where people learn from you or others. What are your thoughts on that, on training people to get better at this?
Yeah, I'm actually training on Twitter. I'm turning I'm turning this into a game. So I have been posting a lot of these duplicated images, and I usually remove the identifiers. I don't want it to make these. I want to just turn it into a game without, you know, the additional
without shaming
severe shaming people. Just making people aware of it. So I'm like, oh, here are 5 panels. Can you spot the duplicates? And I have some examples if people want to play this game. I have some in my presentation, but so I could show them. But it's the main purpose behind this is to make people aware of the problem and to make them into better peer reviewers. If they ever do a peer review, or maybe they become an editor, or they cite a paper, they can look at these things. Because if you're trained to find them, you can see these things sometimes in seconds. So it doesn't add a lot of extra time. And I'm actually, since I've been doing this, I'm getting daily emails from people who say, oh, I found an image and can you look at it? And am I right? What should I do with it? And usually they're sort of excited to have found it, as well as disappointed at the same time, because it's a published paper and so it's out there. So it sort of pollutes science.
Okay, let me ask you a question with one of your I'm going to share my screen because I want to. Can you see it?
Yes, I can see it.
There's just one of your figures. So this is a figure for those that haven't read the paper. This is impact factor of the journal, and then the proportion of papers that Dr. Bik found to have and her colleagues found to have problematic images. So up is bad, down is good. And this is showing that high impact factor journals tend to have less problematic images. Not that surprising, maybe, because it's the referees spend maybe more time looking at them. Not sure, but maybe. Now, the question I have for you here is that I also see this red point up here, and then this purple point here. There's a lot of variability, and I just wonder if you have any thoughts on what's going on here. What explains some of the variability we see within impact factor categories?
Right. So the variability, in my opinion, is explained by the fact that these are different journals that run by different editors. They're run by different publishers, and so each of these group of people have an impact on the quality of images and the quality of papers that gets published. So the one that is at the top there in red, that's a Spandidos published journal, and I don't remember which particular journal that was, but it's a Spandidos is by some regarded to be a predatory publisher, and so it's not
same Hindawi also is
Hindawi. I think Hindawi is actually trying to do better. They have now an ethics research ethics team, and so they're definitely trying to improve things. And but these are papers from older, so they're from 2000 and 2000 and up, most of these papers. So they're a little bit older, and I think a lot of editors didn't realize that this was a problem. So there's other journals who specifically scan for these things, and they have a very low percentage of problematic images.
Interesting. So the next question is from your other paper. This is let me see. I can zoom in so we can all see it. Where to go? This one's I'm sorry about that, everybody. So this is a very interesting one. This is by country of the first and last author.
Mm-hmm.
And first, this is compared to the odds. It's relative to the U.S. So 1 means like the U.S. and then up is worse and down is better. And well, first thing I'm noticing is that it looks like the U.S. is better than the average, because most of these points are above 1. There's a few exceptions. So I'll ask you if you have any comments about that. But then there's 2 that really stand out to me, and it's China and India. So the question is, what is you have, and there's an audience member asking a similar question. What do you think is driving this in these countries? Is it institutional policies or something else?
Yeah, there are sometimes institutional or country-wide incentives to publish. And so, for example, China has had, I think they stopped doing it. They had incentives to publish in international journals. And they also have in a lot of medical schools or medical or hospitals that are not specifically research hospitals, but just clinical hospitals. They have a rule that if you want to get a promotion as a young doctor, if you want to sort of step up to the next level, you have to have a paper published. And these doctors are not given time to do research. And so sometimes they buy papers from a paper mill. So they're maybe fabricated papers. And they just want to have their promotion. They're actually not really interested in research. And so they might do this. And so it's policies that are, I'm sure, written with good intentions. Like we want people from our hospitals to publish, and we want to be a force in international science publications. But these incentives lead to things like that. And we actually, one of the things we did in this paper is to really look at academic culture and incentives, and found that it was associated with that, and not driven by the country, but driven by these policies.
Interesting. Yeah, and then let me be clear that this is not the nationality of the author. It's the institution.
Right.
That's
exactly.
So,
yeah.
Okay. Important distinction. Good. So by the way, you need to take 3 more drinks based on some of the questions I'm seeing here on the.
Oh, okay.
On the Q&A. Okay, I'm going to stop sharing, and then we're almost out of time, so I'll ask you a couple more questions. So this one, this is something that worries me, not specifically about you, but in general, that you in particular, but others as well, have become influential on social media. And a post of yours, although you're very good at not putting out the names and all that, but sometimes people can figure it out. So you have now a bit of power, and you could affect someone's reputation by tweeting their paper as an altered image. So that might concern some people because there's no formal process or anything like that. So and there's not necessarily a way for the authors to defend themselves, although they could go answer on Twitter if they wanted to. But do you have any thoughts on that?
Yeah, I think the work I'm doing is very easy I could easily bring down people or make fun of them, and I try not to do that. So I make a game out of the duplications, but I try to make it about the figures and not about the author. So like you said, I usually don't publish which paper it is, although sometimes I do make exceptions. And there are I see other people immediately yell misconduct or shame. They should jail, put the authors in jail. And I try not to do that. So I try to flag the papers, but it's up to other people to judge. So it should be the institute should do an investigation to see if these what happened, that these images are duplicated, were they fabricated or not. That's the part I don't want to interfere with. I just want to flag these things. But yeah, it's easy to write a blog post and make fun of people, and I try not to do that. Sometimes it is tempting because sometimes you find a lot of dirt on a person. But I try to make it about the papers and not about the people who write these things. I also don't know who of the 10 authors on a paper might have been the one doing the misconduct. All authors are responsible. That this is how science publishing works. You accept responsibility for the data in the paper, but I cannot tell if it was the first or the last or the middle author who photoshopped an image. So it's about papers and not about papers.
Do you email the authors, or is it too much work?
I do that indirectly. If you post on PubPeer, which is a site where you can flag papers and you can say, hey, this figure 1A looks like figure 2B, and here's the illustration. It does send an email to the author. So it's sort of, I don't directly send it to the author because it becomes a, he said, she said type of thing. They'll deny it. But the authors are notified that I posted something on their paper online. And then I'll report it officially by sending an email to the editors, not the authors, but to the editors of the journal, sometimes also the publishers, now that I have a couple of more people who I know are concerned about these things as well. And occasionally, if it's a cluster of things, a cluster of papers from the same author, I will also send an email to the institute, to the research integrity of the officers of the institute.
Wow, interesting. Okay, so and have you noticed? Have you started to see an improvement at all? You've been doing this for about 5 years. Is there any sign of things getting better?
I think yes, things are people are more concerned about it. I mean, it's similar to anything that gets posted on social media and discussed, as we now know with, for example, Black Lives Matter. I think once you bring videos or evidence online and have other people look at it, not just the people who are responsible behind the scenes, but just the general audience, I think collectively people are worried and concerned and demand that these things are addressed. And this is just very general for a lot of injustice in society, but also for science misconduct. I hope by posting these things online, I'm opening the discussion, and I'm making people aware of the conflict of interest that some of these journals have, and institutes as well. Like if there's a very highly influential professor somewhere at an institute that brings in a lot of grant money, an institute might not be very motivated in letting that person go because they're doing misconduct. They might actually be tempted to put things under the rug. And I see a lot of examples like that. And I hope we can change the discussion. And I think it's going to be a very long and slow process, but I do see some signs that things are improving, yes.
Excellent. Yeah. Well, I'll say one thing. You talked about putting stuff on social media, but I gotta say that you do something else that is, I think, important, because social media there could be biases that aren't noted immediately, but you actually did a systematic review and published that as well. I think that's important. I don't think it's enough to just post tweets about problems, but
yes. No, I started by I started by being a serious researcher, and now I'm taking things online. But yeah, I'm very serious in this field, and I hope that I've proven I have a little bit of a track record of publishing papers on this. But now I'm taking, like I said before, I'm frustrated by the lack of response when I did it the official way. And yes, I'm taking now things more and more in the public to make people aware of it.
All right, great. Any parting words for all the data scientists listening?
I don't know. Be honest. Be good science, and make sure that don't walk away from this presentation by thinking that all science is rotten or broken. I still believe that most scientists are super honest and well-willing. They might make mistakes, like doing the wrong statistical test, for example. But I think most scientists are honest and trustworthy. I'm just dealing with a tiny sliver of science that is not the case. Not good science, and but still, I have full faith in science, and I hope you have too.
I agree. Well, thanks again for joining us.
Sure.
You're very welcome. Very interesting, and I'm sure the audience also enjoy themselves. So thanks again.
Okay, thank you. Bye.